welcome to Future Day. It's a conversation with Aubrey de Grey we have here today. It's Aubrey de Grey is the founder and the executive director of SENS, Strategies for Engineered Negligible Senescence organization, and many of you may know what he does. I'll leave it to Aubrey to discuss what SENS is all about later on in the interview here and its relationship to the future and Future Day. But first of all, I might ask you there, Aubrey, what are your thoughts on Future Day? I mean, why have a Future Day?
I think the main reason why Future Day is such a good idea is very much the same reason that, the International Day of anything is a good idea. Namely, it gets the media and the general public across the whole world to actually remember something that might otherwise just be too far down their radar, and to spend even a few minutes just getting themselves back up to speed with why the issue is important. So in the particular case of Future Day, it seems to me that this is a really important thing, really necessary thing to do. It's incredibly easy for most people going about their daily lives just to forget that their daily lives may be very different in the future. And in particular, that their daily lives may be very much better in the future, and therefore that anything that they can do today to make that future come sooner than it otherwise might would be in their interest. That's really, to my mind, what Future Day is all about. It's reawakening people who kind of know what people are doing to develop a better future, but who don't really pay enough attention to it from day to day.
Excellent. Yes, that's a fantastic response. Yeah, I agree with you wholeheartedly. I think we need some sort of ritualistic day where everybody knows a lot of other people are doing the same thing, and so it's socially acceptable to think about. It gives people permission to think about the future, and it's a sort of different in many of these other sort of special days, holidays, or what have you, days of recognition. It is future focused. It's not centered around something that did happen in the past. So it's got that unique aspect to it as well. So I'm wondering, how can we use Future Day to raise awareness and catalyze action in like a general future thinking, or on specific projects, for instance, SENS?
Well, let's talk about the general side first of all. I think that the main way in which an International Day of the Future, just like an International Day of anything, actually succeeds is if the media do actually pick up on it. If you have a big spike relative to your typical day in the number of stories about the topic in question and the prominence of those stories, whether it's on the front page of a newspaper or on the front page of Yahoo, or whatever it might be. I think that certainly applies in this case, and therefore the efforts that need to be happening right now in the week coming up to Future Day need to be focused on that, on contacting, reaching out to the media, so that the most prominent stories, especially of what's happened over the past 12 months since the previous Future Day, the most prominent stories that indicate that the future is coming and that the future is not coming fast enough, can actually be discussed as prominently as possible and get people motivated and activated.
Excellent. And so, what about specific projects then? How can Future Day help raise awareness of those?
Well, let's go one step
Yeah.
in the direction of specificity, rather
Yes.
than talk about, let's talk about one aspect of the future, namely the defeat of aging, which of
hmm
course is what I and Sense Research Foundation work on. Now, I've always been very strong on not just promoting the particular approach to combating aging or a particular organization pursuing a particular approach, but just promoting the whole mission, getting people to really reorient themselves with regard to how they think about aging, get used to the idea, get comfortable with the idea that aging is actually a bad thing, it's a problem, and furthermore it's a potentially soluble problem. And I think really a lot of what Future Day should be about is that really top level 30,000 foot business. Once people have got a bit of momentum in their heads with regard to caring about a particular issue, then it's almost automatic. It's certainly much easier to move them through to looking at specific projects, getting involved in specific projects, and making a contribution. So the first step is, I think, what Future Day is really all about, the more the symbolism, the fact that this is an important thing to be thinking about as a goal and as a feasible goal. And for me, a lot of what we can do to make that happen is to look at the progress that has occurred. Now, one thing that aging has going for it, perhaps to a greater extent than most aspects of radical visionary technology that people like you and me think about, is that it leans on an awful lot of technology that is making strides for other, perhaps simpler reasons. So, for example, in SENS Research Foundation, we focus on applying regenerative medicine to aging. Well, we define regenerative medicine a bit broadly, of course, to include what you might call molecular regenerative medicine, as well as the traditional stuff like stem cells and tissue engineering that you hear about all the time. But the fact is you do hear about it all the time. In the past 12 months, there have been enormous breakthroughs, as there are pretty much every 12 months these days, in getting stem cells to do more, in building artificial organs and such like. One other really enormous plank that the SENS Research Foundation research program builds on, is gene therapy, the ability to manipulate the genomes of people who are already alive. And the ability to do that is still a little way off, no question, but we are enormously closer to it now than we were even 12 months ago, let alone 24 or 36 months, not least because of the enormously amazing technology called CRISPR, which I'm sure that most people of your audience have already heard of, but which is really going leaps and bounds. When it was first announced, it was already semi-miraculous, and it's been better ever since, in terms of both safety and versatility.
Yeah, so for the benefit of those who don't know what CRISPR is, would you like to give it a little rundown and its potential for regenerative medicine?
Yes, yeah. So CRISPR has been called the adaptive immune system of bacteria. And what that means is that it's a system whereby bacteria can incorporate bits of their own infections, what are called bacteriophages, into their own genome as a way of triggering themselves to be able to destroy those sequences when they see them again. And the mechanism by which this material is safely, but nevertheless usefully, incorporated into the bacterial genome has recently been elucidated reasonably well, and we've figured out a way not only to make it work in mammalian cells, including human cells, but also to make it work rather more efficiently and technologically feasibly than it does in its natural environment. The result of that is that people have been able to create genetic modifications to cells much more easily and much more reliably than before. Now, at the beginning, when CRISPR shot to prominence, that was basically the whole story. But since that time, since it's pretty obvious what the potential therapeutic applications are, people have been working really hard to take it further forward. One thing that needed to happen was to make absolutely sure that the genetic modifications that CRISPR would make would be only the desired one, that there would not be what is often called off-target effects. And there's been leaps and bounds. Many labs, including, possibly the single lab that's done the most in this area is that of George Church, a member of our research advisory board, who gave a keynote talk at our conference last August. But other labs also have been doing this. Another thing that's been happening, and this is also, George's lab has been doing this, but many, many other labs as well, is increasing the range of applications to which CRISPR can be applied. Originally, the idea was that it could be used simply to make small genetic modifications, like changing one base pair or a few base pairs at a particular location. And the cool thing was that it could be really relied upon only to change the base pairs at the location in question, the desired location, and that desired location could be customized very easily. The idea of being able to make what are called site-specific modifications to the genome is not new. There are other techniques out there that do that, but those other techniques are much more cumbersome because they require new proteins to be designed for each target sequence. This doesn't require that. It only requires new sequence. All right, so that's great. But then on top of that, there have been additional tricks that have been come up with, even to change RNA, to change the rate at which things get transcribed. One thing that actually I came across quite recently because it's relevant to the way in which we want to rejuvenate the immune system is a way in which one can use the RNAs that a human virus is creating as targets with which to destroy that virus. And this is important because it essentially enhances the ability of our immune system to really eliminate viruses. Some viruses are particularly difficult for the immune system naturally to get rid of. They'll suppress these viruses well, but only to the extent of driving them into a kind of latent, dormant state, which will reactivate periodically. And it turns out that that reactivation plays a big part in why our immune systems don't work so well when we get older. So if we can stop that happening by truly getting rid of these viruses, not even allowing them to survive in the latent form, then it's a big piece of progress. And the group at Stanford just very recently announced a first step towards that, again using CRISPR.
Wow, that's amazing. And so that's a useful technique for people who are still for solving some of the issues related to aging to for people who are alive today. From my reading, earlier reading in CRISPR, it was a beneficial technology to repair maybe some defects in the genome of embryos or like really early stage human development, but wasn't there didn't seem to be much described in the way of actually somatic gene therapy for people who are alive now, but it seems to be the case that this technique can be used both for correcting germline style genes, but also now somatic as well. Is that correct?
You've got it, absolutely.
Okay.
Really, the key thing here is it's all about safety. Gene therapy over the past 20, 25 years has been bubbling under there. You know, people have been working really hard to make it work because it would be terribly useful to be able to modify the genomes of people who are already alive. But the problem is that the viruses that people have been using to deliver these genes and to get them to integrate into our genomes and to do what we want them to do have not been 100% reliable in terms of only doing what we want them to do and not doing other things occasionally. And the thing is, they only have to do the wrong thing very occasionally in order to cause big problems, in particular in order to cause cancer. A lot of the difficulties that gene therapy research has faced over the past 20 years have been around the risk of cancer. Now, CRISPR seems to be escaping that. We at SENS Research Foundation are actually, well, we have developed now actually mice which are potentially able to complement the CRISPR technology, and we hope to be using them in synergy with CRISPR very soon, at least in mice. Our work is doing something which CRISPR can't yet do, namely incorporating large amounts of new DNA into the genome. When I say large, I mean your average way of doing things can only get a few kilobases in. We're talking about hundreds of kilobases. And that's actually a really important thing to be able to do for our purposes, because there are several of the SENS therapies that will need incorporation of new DNA, new enzymes, for example, and we'd like to do them all at the same time. It would be really not very effective if we had to do them one at a time. So, so we'd like to do that, and it turns out that there's another technique using a different type of bacterial technology which we've been able to customize for use in mice, and we hope that in combination with CRISPR it's going to be extremely effective.
Wow, that's amazing. And it's so good that a lot of the SENS techniques seems to be able to work hand in hand with this new developments in CRISPR, and I'm very glad to hear about that. Is there any other progress in SENS that has been that's been made in the last couple of years since last we spoke that you'd like to bring up at this point? It may not have a relationship with CRISPR, but yeah, I'll let you.
We've definitely been accelerating. A number of our projects have been going for several years now, and that's how long it takes to get really significant results. And sure enough, those significant results are now materializing. So around about the time I spoke to you last, I forget whether it was before or after, so I will include this one, we were able to demonstrate that cells in culture could be protected from a molecule called 7-ketocholesterol, which is actually the number one toxic molecule that drives the progression of atherosclerosis, obviously the number one killer in the Western world. We were able to identify some bacteria that could break down 7-ketoglycerol, and then we were able to identify the genes and enzymes that they use to do that. And then the third step, which was the really hard part that we managed to get working 2 years ago, was to modify that gene, so that we could put it into a human cell such that the human cell would be protected. The enzyme would be produced and it would be targeted to the right part of the cell called the lysosome, so that the cell would keep working even in the presence of a quantity of this nasty molecule that would normally be fatal. It was a very clean, decisive result, and we got published in a nice journal. And since then, of course, we've been moving on towards trying to get that to work in other cell types and eventually in mice. It's slow work. It's not research is difficult, but we're getting there. Much more recently, I'm going to say 6 or 8 months ago, we had a big breakthrough, probably our single highest profile paper yet in Journal of Biological Chemistry, where we showed that antibodies could be created which would actually chop up a nasty protein that forms amyloid in the heart. This protein is called transthyretin, and it's famous for causing amyloid at an early age in families that have a mutant form of the gene for it. But in normal people that have the normal form of the gene, nevertheless, in old age, especially in extremely old age, over 100, over 105, it turns out that this particular protein forms amyloid, very like the senile plaques in the brain, but that's a different protein, of course. And this amyloid impairs the function of the heart, and it seems to be responsible for a high proportion of deaths in extremely old age. So it's extremely nice to hear that we've been able to generate antibodies which don't just bind to this stuff, they actually chop it up. They're proteolytic antibodies. And we were able to publish very clear proof of concept of that in JBC about 6 or 8 months ago. Since then, other projects have been moving on. I can't talk about them in such detail because this work hasn't been published yet, but suffice to say that our single longest standing project, which is to make mutations in the mitochondrial DNA harmless by making backup copies of the mitochondrial DNA in the nucleus, that's an area where we've been frustrated for a long time, but we've made very nice progress over the past 6 months or so. And I think I can certainly say at this point that we are a good deal closer to making that really work than anyone's ever been before. So I'm not making any promises about timeframes yet, but I'm pretty happy about how that's going.
Wow, that certainly sounds like an amazing accomplishment. All these breakthroughs you guys at SENS are making are doing things that I can't really begin to describe how important and impactful they'll be today, but also when they're realized. I mean, today, in a sense, it wakes up people to the idea that these sorts of problems aren't insoluble, but also in the future when we can engineer this sort of thing, it's off the scale in terms of the impact, the beneficial impact it'll have on humanity. So I'm very excited about the research you're doing there. Talking about the future, let's talk about the past. Actually, why not? What did regenerative medicine look like, let's say, 35 years ago?
I think it's fair to say that 35 years ago, regenerative medicine didn't exist at all. The beginning of regenerative medicine was probably, well, I mean, of course, you can define that in many ways. One reasonably defensible way to define it would be when Irving Weissman at Stanford discovered hematopoietic stem cells, blood stem cells. And I think that was actually, even that was less than 35 years ago, though I don't quote me on that. But, you know, it took an awfully long time to get even bone marrow transplants to work reasonably well, and we've still got some way to go there. It was a very long time before we discovered other types of stem cell, especially more primitive ones like embryonic stem cells and mesenchymal stem cells in human beings or in other mammals. And we've still got a long way to go before we can really say that we can do exactly what we like with stem cells. The situation with tissue engineering, I would say, is even stronger. Tissue engineering kind of existed 20 years ago, 25 years ago, but it had a kind of false start. People got very excited about the ability to grow, the possibility of growing artificial organs, and actually tissue engineering went commercial rather early in its scientific development. In hindsight, we now appreciate that it went commercial a good deal too early because a lot of people put money in and lost it. But luckily, the science has moved on anyway, and the commercial interest in that area has revived, as it should have done, because the science is ready for it now. So, we're in a very different world now. But yeah, 35 years ago, it was a twinkle in people's eye.
Hmm. Well, there'll probably be a lot of caveats to any answer to such a question about 35 years into the future, but that's the obvious next question. What can we expect in 35 years with what will regenerative medicine look like, and who will benefit from it in 35 years?
Well, one thing that you'll notice from my earlier replies is that advances that are happening in all of these areas are not just making us better at things, but making us better at more things. In other words, it's both the breadth and the depth of this technology that's increasing all the time. And that's rather important to take into account and look into the future, because, of course, what we'd like to be able to do is to apply regenerative medicine to the problems of aging. In other words, to develop what we like to call rejuvenation biotechnologies. Rejuvenation simply being defined as regeneration of damage, repair of damage caused by aging. And it's looking like it's going to happen. I still think that we are on track to develop all of the various SENS technologies that we've been working on for a while within the next couple of decades. I don't think we've been going as fast as we could have done if there had been only science to worry about. In other words, the lack of funding has definitely slowed things down. I would say over the past 10 years, it's probably slowed things down by maybe a factor of three. So we've made only 3 years progress in the past 10 years. But still, that's a good deal better than nothing. I think that if we keep this up, that every step that we make will get people more encouraged about what's going on, increase the credibility and the belief in all of this. And so money will become less and less of a barrier. So I would say that at this point, I think we've got at least a 50-50 chance of developing all of these technologies and bringing aging truly under the control of regenerative medicine and its associated technologies within the next 20 or 25 years. Now, as I've always said, even from 10 years ago, that is an extraordinarily speculative time frame prediction. I certainly think there's at least a 10% chance that we won't get there for 100 years if we hit a million different problems that we haven't thought of and encountered yet. But still, so what really? A 50-50 chance is quite enough to be worth fighting for.
Absolutely. Definitely worth working towards. Well, okay, so Future Day is often thought of as, a celebration. It's also about education and about increasing the economy of attention on the importance of foresight. So, yeah, what can we do to integrate critical thinking about matters of import, strategic forecasting into education, for instance, primary school or high school?
Well, I think there's two questions there, really, or at least two answers to the question.
Okay.
I think that we want to incorporate good critical thinking into education to a greater extent, simply actually for a very broad range of reasons. Ultimately, if you're not educated in how to think critically and rationally, you're not going to be able to do a whole bunch of things that go way beyond visionary, distant future technology. You won't be a good scientist at all, you won't be a good engineer, you won't be a good many things. And certainly there's an enormous shortfall there. Teaching is far too underfunded, to be honest, and that means that teachers are not taught well enough how to teach, and there's not enough quality teaching out there and quality time spent teaching not just facts, but methods, and in particular methods of how to think. But more narrowly towards Future Day, I think we need to be thinking not only about how to get people to think rationally, but also how to extrapolate. There's an enormous hindrance in society to future progress, which is fear of the unknown. And of course, fear of the unknown is, in a very real sense, perfectly justified and rational. We are aware that some risks that can't be evaluated should just be avoided, and because we just want to play it safe. I don't think there's anything really wrong with that. But it can be taken too far. And certainly in the case of aging, it is taken too far by the overwhelming majority of society. Why do I say too far? Simply, I say that the overwhelming majority of society do not apply a sense of proportion to the question of aging. They will say, oh, well, if we were to defeat aging, we might create these new problems. Or they might equally say, if we allocate resources to trying to defeat aging, that will take resources away from things like defeating malaria in sub-Saharan Africa and so on. And of course, these things are true, but people kind of stop there and they don't go on and do the absolutely vital next step of asking how true these things are. How much would the amount of resources available to treat malaria in sub-Saharan Africa actually be reduced by adding an extra zero to the amount of money put into trying to defeat aging? The answer is probably infinitesimally, simply because the money comes from different places and people fund it for different reasons and so on. If we ask the question, what about these problems that might be created? People are very, very prone to come up with potential problems and then immediately to shut their brains off and not actually think about the opportunities that might be available to avoid those problems or to solve them, or even the possibility that the problem might actually occur and not be soluble, but it might be considered to be the lesser of two evils relative to the problem we have today of 100,000 people a day dying after a long period of decline and decrepitude and disease and general misery. So, to my mind, that's the kind of critical and rational thinking that is overwhelmingly most seriously missing, at least insofar as the topic is future day.
Excellent. Well, it's been wonderful to chat to you again, Aubrey. If there's any concluding remarks about the future or the future day or SENS that you'd like to impart at this final stage of the interview, please do so. We still got couple of minutes left.
Yeah, sure. I think I just would like to say that, first of all, I want to congratulate you and many of your colleagues for coming together and creating Future Day in the first place. It's not something that I managed to spearhead, so I'm very pleased to be joining the crowd of people, especially prominent people, who are supporting this. And I also want to say that I will be tracking it afterwards, and I think everyone needs to track it. You don't need to just get online and read the media and so on the day itself, but actually try to maintain momentum after that. Everyone who is, in general, supportive of visionary technology being pursued more aggressively and to the greater benefit of humanity has a kind of duty to actually do that, to actually not just one day a year, but actually every day of the year to make a contribution to maintaining and increasing momentum, increasing resources that are available to this, and just generally making it happen sooner.
Excellent. Well, it's been a pleasure, and I'm certainly tracking what you've been doing there at SENS. I look forward to going to another one of these SENS parties that we had in 2012. That was certainly a lot of fun. So I'll be putting links to SENS, and I'll be doing a little bit of research on the topics you brought up and providing links to those in the description. But thanks so much for being part of Future Day, Aubrey. It's been a real pleasure, and I certainly do, for my own vested interests, and everybody, wish you all the best in future development at SENS. So thank you so much. It's been a wonderful experience chatting with you today. Cheers.
Thank you. Bye-bye.